Good afternoon. My task over the next few minutes is to explain how we have used the platform of PubMLST to support a new research network called the IRIS Initiative. Now, we set up the IRIS Initiative in the midst of the COVID pandemic. So the COVID pandemic has affected the entire world, and every country has implemented some sort of containment measures to try to block the transmission and spread of the coronavirus. So some of us were having discussions around what this might mean for invasive bacterial infections. And one possibility was that we would see an increase in invasive bacterial disease because of this recognized link between viral infections and subsequent bacterial infections. However, we might also expect to see less invasive disease because of the containment measures that were put into place for COVID-19 and the fact that that would be not just reducing transmission of a viral pathogen, but also bacterial pathogens. And then subsequent to that, if in fact we were seeing a reduction in disease, invasive disease, we might expect then that there would be perturbations in invasive disease as containment measures were lifted. So we set up the Invasive Respiratory Infection Surveillance Initiative, or the IRIS Initiative, with a single aim: to assess changes as quickly as possible in the incidence of invasive disease caused by the three major bacterial pathogens, Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitidis, and then as a comparator organism, Streptococcus agalactiae. And to do this, we assessed changes in what were being submitted through routine surveillance systems in national reference laboratories. So we were not setting up a new study in terms of the collection of the data, as in the isolates that were being reported to the reference laboratories, but we simply used data that were already part of national surveillance programs. We initially contacted people within our network of collaborators and scientific colleagues within Europe, and then we quickly expanded that network to other countries. So you can see on this map that we have a large number of European countries who are involved in IRIS, but we also have Canada, Brazil, South Africa, Israel, New Zealand, and then South Korea, China, and Hong Kong involved as well, so that we were able to get quite a good perspective on what was happening globally across 26 different countries. And it's at this point I'd like to say that we've just submitted our first manuscript for publication, and there are 95 co-authors on this paper. So we have a large number of collaborators on this project, and every one of whom is really valuable to this project in terms of collection of isolates, characterization of isolates, collection and submission of the data, and then analysis of the data that we have now in IRIS. So many thanks to everyone for the work that has gone into this project already in a very short period of time. Now, part of the reason that IRIS was set up quite quickly is that we aim to keep things very simple right from the start. So the first point is that the IRIS database is actually part of PubMLST. So PubMLST provides the structure and provides the resource such that we could set up a database very quickly and very easily. Secondly, we focused on minimal data to start. So we were only asking laboratories, who were of course very stretched by COVID-19 at this point in time, basically just asked for minimal data. So could they report the cases of invasive disease and the date of sampling of the specimen to start off initially, and then we could add more data and update the database later. And most laboratories did that. There were quite a few laboratories that have in fact submitted quite a lot of metadata. So we're now starting to work on that data update phase. Furthermore, we were able to set this up such that IRIS participants could enter the data themselves. So the database is a private database, and IRIS participants need to log in to enter data, but they can do that directly from their laboratory to our database, or they can send the data to us and we can upload those data. Now, data protection is really important for this study because we're dealing with laboratory-based data. So the IRIS database is a private database, and it's only accessible to those who are participating in IRIS, and they need to log in with a secure login and password. Importantly, the laboratory who is submitting data to IRIS also retains ownership of their data. So this is not a study where a laboratory will submit data to a database and then someone else goes off and analyzes those data. This is very, very much a partnership amongst all of the reference laboratories. And finally, just to say that we don't collect any patient data, so we don't have issues around that. So IRIS, as I say, is hosted on PubMLST, and we use the PubMLST platform. What you see here is a screenshot of the sort of front page of the database. This is public, but then, as I say, users need to log in to get access to the individual databases, one of which for each of the different bacterial organisms. What you see here is a screenshot of the German data. This is a drop-down menu, so we can look at individual countries, or we can look at the aggregate for any one of these 4 pathogens. And so what you see here is a cumulative curve, number of isolates on the y-axis, and then the week of the year across the x-axis. And you can see that 2018 and 2019 data look very, very similar. They're in blue and orange. And you can see immediately that in 2020, there was this rapid reduction in invasive disease. This is pneumococcal data, starting around this period of time. Now, if we look at all of the data, and I'll just show you one figure from our paper that we've just submitted, but this is one of the headline figures, that what we found is that invasive diseases, in fact, plummeted in early 2020. So what we see here are cumulative curve graphs for each of the 4 organisms that we've assessed. If we look at the pneumococcal data, we have nearly 63,000 isolate records. We're again looking at a cumulative isolate count on the y-axis, and we're looking at the week of the year on the x-axis. This red line indicates week 11, which is when the WHO declared the COVID-19 to be a pandemic. And what you see for pneumococcus, as you can see for both Haemophilus and meningococcus, that in 2018 and 2019, the data are virtually superposable for pneumococcus. In other words, the number of isolates that were collected each year cumulatively over 2018 and 2019, and how much that differs with that now 2020 data. And you can see that this inflection point is right around the time that the COVID pandemic was declared. In the paper, we've actually taken this one step further, and we have assessed the containment measures that each country has implemented in response to COVID. And we've considered the stringency of those containment measures using data from the Oxford Blavatnik School of Government. We've also assessed Google mobility data to assess how populations were moving around, as in spending less time in workplaces and more time at home. And we have models that explain how we get to this sort of reduction in disease. But the message here is really that we have data for these 3 different pathogens, which look very, very similar, this reduction in disease around the time of the pandemic being declared. And one criticism that came up with these data, or what could be criticized about these data, is that perhaps it was just that the laboratories were so overwhelmed with COVID that in fact they weren't receiving the number of isolates that they would expect to receive in a normal year. Now, every laboratory said they didn't feel that that was the case, that they felt that things were proceeding as normal. But to assess that a bit more stringently, we also looked at the Group B strep data. Now, Group B strep is not a respiratory pathogen. These are invasive isolates from 9 different IRIS laboratories who routinely collect Group B strep data. And you can see the pattern here is very different, that there's really no difference between any one year, 2020 versus 2018 or 2019, in terms of the submission of Streptococcus agalactiae invasive data. So we think this demonstrates that in fact this is not a problem with receipt of samples, but in fact these data are real, and that we would hypothesize that the reduction in transmission of bacterial pathogens coincided with the containment measures that were implemented as a result of COVID. So to summarize, we very quickly set up the IRIS initiative in response to the COVID-19 pandemic. And we were able to do that because we had the PubMLST platform on which to set up the database and facilitate this data collection, not just by uploading the data ourselves, but also by allowing IRIS participants to be able to upload their data directly. This is very much a collaboration. It's very much an international network and a partnership amongst all 26 countries and all of the laboratories that are involved in this work. We have just submitted the first manuscript, as I mentioned, and it remains to thank everyone who is involved in IRIS for what has been an extraordinary amount of effort in quite a short period of time, and in fact, quite an enjoyable one. It's a very congenial network, and I think that has been a great deal of fun for everyone who's been involved. So thank you very much, and I'm happy to take questions.